Now we will start with the study of artificial hybridization. Artificial hybridization is a specialized process in which desired pollen grains through the process of pollination are interacted with the stigma of the other flower Thus, it is a process of pollination in which the desired pollen grains can be obtained by interacting with the stigma of the other flower and by avoiding its interaction with other unwanted pollen. By this method, various hybrids can be produced in an artificial manner. Now the process of artificial hybridization takes place mainly in form of three steps. Thus, the three steps or processes which are involved within it are as follows. First, selection of the parental source, that is the plant which has to produce hybrid. Parental source involves both the male and female source, that is the male and female plant that has to be interacted. To produce hybrid. Then the second process following it is known as is emasculation and bagging. Emasculation and bagging are the techniques in which the female parent and the anther is split off. That is, the male and female parent are separated from each other. In bisexual flowers, this technique is generally used for bisexual flowers. In such flowers, the anther, that is the male parent, is separated by using forceps, that is the equipment like forceps, from the stigma of the flower, that is the female parent. This separation, that is the separation of the anther from the stigma of the same flower, should be carried out very carefully, that is, should be carried out without any injury. This process of separation of the male parent from the female one is known as emasculation. Thus, the process of separation of anther containing pollen that is the male parent from the stigma of the same flower or of different flower but of the same plant that is the female parent is known as emasculation. The following emasculation or technique known as bagging technique occurs. In this, the emasculated flowers that is the flowers which are separated from the anther are placed in a bag-like structure that is made up of butter paper. These emasculated flowers are placed in such a bag-like structure in order to avoid its interaction with the unwanted pollens. Because such emasculated flowers only possessing female parent are now sensitive and can interact easily with the unwanted pollens and get contaminated. So, this interaction that is avoided with unwanted pollens also avoids the contamination of the stigma. This technique is called bagging technique. Following bagging technique, when the pollen grains, that is the mature pollen grains of the other flower mature pollen grains or mature pollens from the other flower is collected and the stigma that is placed within a bag gets receptive, then the third technique, that is third process, cross-pollination occurs. Thus, in this step of cross-pollination, the mature pollen grains of the other flower is interacted or pollinated with the stigma of the other flower, and in this manner the cross-pollination is executed. This was all about the bisexual flowers. But in unisexual flowers, since male and female organs of the plant are both separated from each other and are present on different flowers, no emasculation of the flowers is needed. But yes, however, in order to avoid the interaction of the stigma with other unwanted pollens, bagging technique is necessary, which is further followed same as that of bisexual flowers, that is cross-pollination. This is the only difference between unisexual and bisexual flowers. The rest of the process of artificial hybridization remains the same. Thus, in both unisexual and bisexual flowers, after the process of cross-pollination, these stigma of the flower are then again rebagged. Because still these stigma are sensitive to the environment, and so in order to avoid its contamination, they are repacked for some time. And thereby gradually with time the fruit develops out of it. To produce hybrids. In this manner, the process of artificial hybridization occurs.